Good morning to all. A warm welcome to all the participants of today's 9th webinar of Faculty of Pharmacy, an international webinar organized by Faculty of Pharmacy, Dr.M.G.R. Educational and Research Institute on current trends in next generation gene sequencing by Mr. Shaun Seonwoo, Assistant Sales Manager, NGS Division, Macrogen, Seoul, Korea. Faculty of Pharmacy, Dr.M.G.R. Educational and Research Institute has been successfully organizing series of national and international webinars and FDP with eminent speakers from all over the globe with a vision to aggrandize the intellectual wealth to the students, faculty, research scholar, industrialists, and pharmacists. On this juncture, I would like to gist on today's webinar, which involves one of the most innovative and fast-moving fields in healthcare, that is genomics, which encompasses the sequencing and analysis of human genome. Genomics is enabling pharmaceutical companies to take a more personalized approach to drug development and will lead to safer and more effective drugs. Thank you and have a happy learning. May I now request our principal, Dr. N. Harikrishnan sir, to deliver the welcome address. Sir. Excuse me, sir. Sir, we are not able to hear your voice, sir. Sir, we are not able to hear your voice, sir. Yes, sir, not audible, sir, not audible. No, sir, not audible. एक मिनट सर एक मिनट सर
Sorry. So good morning to all the participants. So after a successful conduction of the webinar series and online program on behalf of Faculty of Pharmacy, Dr.M.G.R. Educational and Research Institute, welcome all the participants to the international webinar on use of current generation gene sequencing technology. So in this moment, I would like to dedicate my heartfelt thanks and gratitude to our founder chancellor, Dr. Asst. Seonwoo sir, Honorable President ACS Arun Kumar sir, and Honorable Secretary through A. Ravikumar sir for permitting us to organize this webinar and their support for the Faculty of Pharmacy. So here in this juncture, I also extend my sincere regards to our Vice Chancellor, Dr. S. Geetalakshmi, and the Registrar, Dr. C.B. Paranivelu, for their motivation and guidance. I would like to render my thanks to Mr. Shaun for accepting my invitation, and also here I want to express my extended thanks to acknowledgement to Dr. Sendil Raja for arranging an excellent webinar, for arranging a second webinar. And also I extend my sincere acknowledgement to Dr. Hemalata and even coordinator Mrs. Komal and other faculty team members. So all the team members, really my sincere thanks to all the faculty and my team. So I hope all the participants will get a deep understanding on generation gene sequencing technique and its application and the importance through this webinar. So thank you very much for all the participants. So really this is going to be a nice webinar. Please all the participants, while they're taking lecture, please unmute your mics This is from the host. So thank you very much for giving the opportunity. So thank you, sir. Thank you. Thank you, Komal.
Thank you, sir. May I now request Mrs. Dhanlakshmi ma'am to give an introduction about today's speaker. Ma'am.
Good morning to one and all. This is Dhanalakshmi, Assistant Professor, Faculty of Pharmacy. It's my greater pleasure to give key introduction about our today's chief guest, Shaun Lee. Sir is currently working as Assistant Sales Manager in his division, Macrogen, Korea. Sir having the academic excellence in Bachelor's of Science in Biological Science and completed in the year 2011, and Sir completed Master of Science in Biological Science at the year 2017, and Sir having work experience in Republic of Korea Army as research personnel in the area of genetics from the year 2013 to 2016. And Sir worked as the Assistant Manager in Excellence Incorporation, Yongjin, South Korea, under the Department of ELISA from the year 2018 to 2019. And Sir published in the thirst area genetics and having the publications entitled with multiple gene polymorphism can improve the predictions of non-orbital fractures in post-menopausal women at the year 2013 and genetic epidemic H3K27 modifier genes at the year 2018. It's my greater pleasure to give a key introduction, sir. A warm welcome to you, sir. Thank you, ma'am. Ma'am, the forum is to you, ma'am. Komal, ma'am.
Thank you, ma'am. Let's begin today's technical session by our esteemed speaker. I request all the participants to mute themselves during the technical talk and pin the speaker's presentation. Participants are requested not to present themselves during the talk. Participants can type their queries in the chat box, which will be clarified after the talk. May I now request the speaker of today's webinar to start the talk. Sir, please.
Thank you very much. Okay, I wasn't expecting that this kind of real formal and great introduction, but thank you so much for everything. So, yes, please, take a comfortable position and enjoy the webinar. I'll be starting right away. So, I'm just going to shut down my cameras because I cannot.
One of Macrogen's pioneering technologies, the whole exome sequencing. It can be used to reveal various types of genetic mutations that cause diseases in certain parts of the human body. Exome sequencing is a cost-effective way to selectively analyze the exome regions of the DNA code for amino acids. Moreover, the targeted sequencing service enables an even more accurate analysis of genetic mutations by reading only the disease-associated gene regions. Through transcriptome sequencing, mRNA expression can be precisely measured, and new information about novel transcriptomes and fusion genes can be acquired. Countless microorganisms exist all around us. Metagenome sequencing analyzes microbial populations in various environments. The discovery and research of previously unknown microbial genomes is measured certain gene expressions affected by environmental factors. Macrogen has been accumulating global genomic data by putting continuous effort into research and development and publishing numerous papers. Now, as we propose solutions to diseases based on genomic data, we at Macrogen believe we can together create healthy and happy lives for mankind. This is Macrogen, where we explore and discuss all kinds of genomic information.
Thank you for watching, everyone. Now, as it was shown in the introduction, our company is currently the world leader in terms of research sequencing market. And we also have the world's 5th largest gene analysis capabilities with over 30 petabytes of big data analysis capabilities. And although the video only showed NGS, we're also into CDS, clinical diagnostics, direct-to-consumer or patient genomic sequencing, microarrays, oligos, or genetically engineered mouse. We also have a wide network of global subsidiaries and branches, under which we also provide multiple genomic sequencing services. And we also have over 118 papers published in SCI journals, of which 16 papers are published in Nature. So if you ever have genomic sequencing or any sort of genomic analysis needed, please be sure to find Macrogen. We'll always be there as your partner. Now, that's enough advertisement. Let's get started with the actual presentation now. Okay, in order to understand the world of sequencing, we have to understand a bit about the genome. So let's begin with the basics. The genome is a word that's combined from the word gene and chromosomes, and it is the complete set of genetic material present in a cell or organism. Usually this is in the form of DNA, and they are usually consist of thymine, adenine, guanine, and cytosine, these 4 nucleotide bases, which form a unique code which are translated into various proteins that build our body. So, in order to go further, let us talk a little bit about the central dogma of molecular biology. In one sentence, central dogma of molecular biology is that DNA makes RNA and RNA makes protein. So if there's this strip of DNA sequence in the genome, this will get translated into this sequence of transcribed into this sequence of RNA, and that will be translated into this sequence of amino acids or protein. And once the proteins are made, these will eventually form the body of an organism. So in essence, if you want to study molecular biology, you need to study the genome. And so how do you study a genome then? Usually the most well-made analogy of a genome is a book. If a human genome is a book which contains instructions for making protein, which in turn constructs the body, it will contain 23 chapters represented by chromosomes, and each chapter will contain 48 to 250 million letters, A, C, G, T, without spaces. So it will contain over 3.2 billion letters in this book. So as you can see, a book consists of letters, and the question is, how do you learn what is in a book then? The answer, quite simple. You have to read the book. So this is the process of sequencing. Sequencing is the act of reading the book. And in order to read the book, you need technology to do so. And so in order to solve the mysteries of genome, a number of worldwide companies have dug in to make, why is this, excuse me? Okay, pardon me. So a number of sequencing companies have been developing technologies. These are just a few examples, of which Illumina and Pacific Biosciences are the market leaders in this field. Now, I'm not going to go too deep into the technologies of the companies. Suffice it to say that Illumina technology focuses on creating a large number of sequencing reads to read a large genome in parallel, while Pacific Bioscience Technologies more focuses on creating long reads, which allows more accurate reading of the genome. So I'll be explaining that in details in a moment. So currently, there are two mainstream sequencing technologies in the world. The first generation sequencing technology called CES, or capillary electrophoresis sequencing, was commercialized in 1995, and it can read about 500 to 800 base pairs of data for each run. And it takes about 2 hours of reaction time. So using the same analogy of the book I used a while ago, you can say that this technology, each time it reads, can read 500 to 800 letters in a book. However, this is way too slow to read an entire book that's over 3 billion letters. So what came out is the second generation technology, NGS. Now, this was commercialized in 2005, and unlike CES, it can read about 3,000 gigabases of data, or 3,000 giga letters, each time it is read in about 1 to 6 days of reaction time. This truly has revolutionized the world of sequencing because it allowed us to read huge amount of data in a very short time. So let me go into a little detail of how this is sequenced. In the next generation sequencing, the workflow is as it's shown in this table. You first gather samples from nature. Usually, you'll have to extract DNA or RNA from whatever samples of biological organisms you get. And after that, you have to construct what is called libraries, which are modified RNA or DNA molecules, which can be used for the platforms, the sequencing platforms, to actually read each sequence in this fragments. And after the sequencing is done, you have to do bioinformatical analysis. And finally, you get whatever data is in the genomic sequence. In a more visualized example, this is how an Illumina technology would work. Assume there is an unidentified genomic DNA here, and you want to know what sort of sequence it's making. What it does is it fragments each genomic DNA sequence into fragments smaller than 600 base pairs, and these are constructed into libraries. After that, the libraries are sequenced with each of the libraries being read by about 300 base pairs or shorter at a time. And after they are all read, the overlapping DNA sequences are matched bioinformatically to get an assembled sequence, as in this example. So this would be a completely sequenced and assembled genomic DNA. And that's how basic sequencing technology works. Now, as I said previously, next generation sequencing has revolutionized the world of sequencing because it can basically sequence so much that it can be sequenced using a first generation sequencer of CES. So if you use the first generation sequencing technology to sequence an entire human genome, it would take 42 years. But using NGS sequencing, you can do it in 1 week. So that reduces the cost of sequencing, at the same time increase the speed immensely, which has allowed a new field of systemic biology and big data science to emerge. If you can see here, sequencing has revolutionized, sequencing revolution by NGS has dramatically reduced the cost of the sequencing per gigabase. So in 2003, when the first whole genome sequencing was completed, it cost about $3 billion and 13 years to do it. In 2019, you could even finish the whole genome sequencing in about $600 in 2 days of time. So that sums up to that one sentence: you can sequence anything, anytime. And this sequencing technology not only is being has revolutionized the world of sequencing, but it is also constantly evolving into even better sequencing. Pardon me, you said something? Nothing? Okay. So it's evolving into getting even longer reads, or for instance, Illumina could only read 300 base pairs at a time, but PacBio was allowed 20,000 base pairs. Or it's also evolving into involving even smaller samples to sequence. So instead of a bulk sample, you can sequence a single cell RNA, or even smaller samples like exosome RNA, which will be explained in the next few slides. So with all this background in mind, let's move on to what applications NGS can provide. Now, because of advanced technology, NGS can be applied into so many different fields. Oh, excuse me. Someone has interrupted my. May I resume? Okay. Okay. May I start? Okay, good. Okay.
Yes, sir.
Okay, I'm having positive feedback. Okay, thank you. So there are diverse applications of NGS available. So even from the smallest single cell, RNA, the DNA molecules obtained from this can be used for whole genome sequencing, also exome sequencing, methyl sequencing, and chip sequencing. And once after it's done with transcription, it can be used for RNA sequencing, including total RNA sequencing, mRNA sequencing, and small RNA sequencing. So basically, you can sequence anything now. And the genome sequencing can be divided into these categories depending on several factors, usually the availability of reference genomes. So the first thing I'm going to explain is DNA sequencing or genome sequencing. Everything begins by studying the genome. And the first thing you have to do is construct a reference genome. These are just some of the papers that was published in some of the greatest journals in the world, Nature and Science. And these all talk about how the genome of certain animal was sequenced and assembled in full. So there's chicks, horses, cats, and pigs. And what this de novo whole genome sequencing does is it creates a reference genome which can be used for further applications. Now, and why do you need a reference genome then? Like I told you in the video, because every genome is actually very diverse. It can contain so many variations, including nucleotide variations, structural variations, of which nucleotide variations include single nucleotide polymorphisms, insertion and deletions, copy number variations, and structural variations. Now, all these variations in the same organism causes immense difference in the genotype and the phenotype. For instance, if this is a reference genome, a genome with SNPs would actually be slightly different from the reference genome, but with addition of insertions and deletions, that could be even more different. And when it comes to copy number variations, even more different. So genetic variations change the genomes and the phenotypes alike. So in order to study these changes using the reference genomes, you have to either do genome sequencing, exome sequencing, or targeted sequencing. These are all the same tools to study the whole genome, but the difference is that in a whole genome sequencing, you literally sequence the entire DNA. So usually the cost is higher, and at the same time, it cannot get enough sequencing depth with the same amount of data. So what comes out later is a more targeted sequencing approaches such as whole exome sequencing, which attempts to sequence only the exome. By getting the same amount of data, they can get much more in-depth knowledge of the genome of the targeted regions, and which can even go even deeper by using targeted sequencing, which goes only deeply into individual short sequences in the whole genome, giving you even more opportunity to much lower cost. Now, but the genome sequencing is just a genome sequencing is just the basis of all studies. But what's more interesting to scientists nowadays is the transcriptome sequencing, which includes de novo transcriptome assembly and RNA sequencing of various RNAs. Now, as I told you in the background section, everything written in a genome has to be transcribed to RNA to become proteins. So by studying the RNA, which is very, very abundant, researchers can learn more about how proteins are created and eventually how it affects the construction of our body. So, transcription sequencing attempts sequencing of all sorts of RNA, starting from mRNA, which is the coding RNA that gets translated into proteins, or non-RNAs, which affect. Excuse me, may I continue? Yes, I'll take that as a yes. And so, okay, so these sort of different types of RNA can always be sequenced, and using these sequenced RNA, a lot of studies can be done, including quantification of gene expression by counting the number of mRNAs that have been expressed, or discovery of novel transcript and fusion genes, and even alternative splice transcripts and single nucleotide variants in the RNA. So, using the data, a scientist can, for instance, compare the genetic expression of a gene in several different samples, or using them, compare the expression levels in different targets and how it changes over time. And depending on using the expression data, they can also construct several different maps, which gives them combination of what sort of RNA is expressed under which situation. So all these research can be done, but the most advanced technology of here is called the single cell RNA sequencing, which was considered the breakthrough of the year in Science and Nature in 2018. And what it does is, as everyone knows, our body's tissues is not a single cell. It is a combination of multiple cells which have very different characteristics. For instance, if this retina tissue is there, it could contain rod cells, pigment epitheliums, horizontal cells, microglia, bipolar cells, ganglion cells. And all of these have different characteristics and they express different genes in them. So if you just do standard bulk RNA sequencing, you have to sequence the RNA as a whole, which will effectively become a combination of all RNA in the sample, all like a shake. But in a single cell RNA sequencing, it's going to be a salad in which you can actually tell which RNA comes from which organism, which cell. So, for instance, if you look at the differential expression of a certain gene in an optic nerve and compare that with optic nerve in eyes with glaucoma, the differential expression will show as these in different samples. However, these are total RNA sequencing and not single cell sequencing. And if you actually differentiate the single cells into multiple different layers, you can actually get the genetic expression of certain genes in different cells. And as in this example, you can see that gene number 1 is expressed highly in photoreceptor cells, but using a drug that targets that average clone, you get to destroy that particular cell. But at the same time, the other cells who do not react to these drugs will not have much effect to the same drug. But by doing the single cell RNA analysis, you will be able to discover different biomarkers in the cells and develop precision drugs according to them and be able to cure a disease directly using these data. And also, several of the other profilings are available using biomarkers of pre- and post-treatment analysis using how does the treatment affect certain cells you're targeting. And by getting the biomarkers of the cells before treatment of drugs or after the treatment of drugs, you'll be able to screen the effect of your drugs and how effectively they can have effect on the cells, and all at the same time can screen out the cells that do not react to drug therapies. So this is how genomics is intertwined with the field of pharmacology or drug discovery, but it doesn't end with just the single cells. Now the world is moving on even further into this world of exomes. Now, exomes are vesicles excreted by cells that affect other cells by secreting molecules in them to various other cells affected by the exome. For instance, cancer cells will be excreting several exomes to other cells and signaling them to change into cancer cells, as in this video here. And also the exomes can be used pharmacologically, where exomes to target the cancer cells to die and fix. So even the exomes are being used very commonly in the field of drug discovery now. Okay, the video was in the wrong sequence. So these sort of exomes are continuously being studied as new drug targets and how these can be exploited to heal diseases, cure diseases. So when an exome is excreted, it's going to get uptake by other cells by endocytosis and other sorts of direct fusion or any sort of ligand binding. And these cell-derived vesicles called extracellular vesicles, exomes, contain DNAs and RNAs and proteins. And these are present in various biological fluids, including blood, serum, plasma, and urine. And these exomes play very key roles in intracellular signaling and several potential roles in disease prognosis, therapy, and biomarkers for disease. Now, because an exome contains so many different molecules inside, including non-coding RNA, mRNA, and pre-mRNAs, it can be used as targets for medicine, vaccine, diagnostics, cosmetics, drug delivery, and all sorts of clinical studies. So again, even in the study of exomes, the key to it is to study what is in it. And in many cases, those are RNA. So sequencing RNA in these small exomes are very key to studying drug effects and treatment prognosis. So this is the field of the RNA sequencing being used in the field. And there is also the field of epigenome. Okay, sorry, this somehow got... Okay. Where is... Okay, sorry, just ignore this slide. The next is metagenome sequencing. Now, metagenome sequencing is about studying the metagenome, or the collective bacterial population in an environmental sample. So, the genome of microorganisms from an environmental sample has numerous applications. What kind of microorganisms live in the sample? How do they interact with each other? And what do they do to the environment are one of the key questions asked by metagenome sequencing. So several of projects exist to database these microbiomes of environmental samples, including HMP, MetaHit, EMP, and Teragenomes are just to name a few. And these microorganisms affect the environment and the world significantly. For instance, microorganisms are climate change indicators which can react very sensitively to climate change. So, and also microorganisms can be used for air pollution control, fermentation for good food, and even antibiotics, which are all created by microorganisms. And again, these microorganisms have DNA of their own, and these individual DNA cannot be sequenced one by one for sake of cost. So metagenomics allows you to study these as a whole in the field. And also, metagenomics can be applied to humans in terms of human body. Now, human body is a large system that's full of microorganisms, and they affect us day and night. Several organisms in the number of 100 trillion cells and over 3 million genes exist in our body. And these don't just stay in our body, but they're also constantly reacting with the environment and also affecting our health status very strongly, which can be a huge plus side to the field of pharmaceutical industry. So in a human, for instance, human intestines are full of a diverse microorganism environment. And this can strongly affect several conditions of our body, including obesity. Like, for instance, in many obese people, it has been discovered that there is very low microorganism diversity in the intestines. And many scientists have studied this and starting to gain evidence that the microorganism diversity can strongly affect bodily conditions like obesity and even depression. Although so far it has not been studied in humanity, several mouse studies show that injection of microorganisms in the mouse improves depression. And again, these sort of metagenomics in the field of pharmacology requires to have its genome studied in advance before they can go into any further studies. And lastly, there is also the field of epigenome, which is the field of studying gene expressions that are not affected by the actual DNA sequence changes. So Epigenome is the study of how gene expressions are regulated by not having effect on the actual DNA sequence itself. So in this example of monozygotic twins, they share a common genotype and they look genetically identical when they're young. But as they grow up, the two develop very different mental disorders and even cancer. And several of these factors are affected by the environment. And the environment is known to actually not affect the actual DNA sequence, but the DNA methylation, which is a DNA modification on the level of okay, am I back? Okay, I'm back. So study of methylations on the DNA can show various changes that in some 3-year-old twins, the two have similar methylation patterns, but 50 years, they show very different methylation patterns or acetylation patterns, which in turn can be actual cause of these several mental or cancers they're suffering from. And several studies can be done to study these sort of differences, which include bisulfite sequencing, reduced representation bisulfite sequencing, and chromatin immunoprecipitation sequencing. And these sort of technologies all in common study the methylation patterns of the DNA without actually changing the DNA sequence itself. So these sort of sequencing technologies are applied as very basics of all studies needed to improve medicine and pharmaceutics. And these studies are continuously improving our lives, and even as of now, not just in the lab. And more importantly, it is also being directly used in the field of healthcare without actually, it's already leaving the lab and it is entering the healthcare market even as we speak to help people live better lives. So here is the field it's developing most rapidly thanks to the development of genetics. Now, as in the previous slides, every person has a different genomic composition. So if there's a large number of cancer patients, for instance, if they have cancer, by using the same therapy and all of them will actually have varying effects. Some will be effective, some will actually have no effect, and some might even have adverse effects. But by sequencing the genome and learning their genetic compositions before you study them, you will get information on various biomarkers which can be used to give targeted therapy to each of the different individuals, which will give much better effect in them as a whole. This field is called precision medicine. And again, it is a most well-studied field in medicine and healthcare. And also, genomics data is now also allowing personalized healthcare in various fields, including our own lives these days. So the study of cancer, pharmacy, reproductive, personalized, and second genomics, all of these moving on directions to giving actual benefit in real life rather than just in the lab. And Macrogen has also launched several of these services called personalized genetic service. So in a person's life, several factors could affect their physical conditions and diseases. Some could be lifestyle, some could be environmental factors, but genetic factors are unchangeable. So what the world is now doing is trying to learn more of the genetic factors of individuals, and using that data, many companies, including Macrogen, is proposing several environmental or lifestyle solutions to clients so they can actually live a better life based on their genomic data, their genomics data, and also using their metagenome data in their intestines. Some metagenome or bacterial composition in the intestine could affect your lives in a numerous ways. So what we do is we study the several different species known to affect human health in our intestines. When we provide healthcare benefits to our clients, telling what sort of microorganisms are living inside you and how this could be exploited to make you live a better life. And these is the field called personalized genomics. And several companies in the world are servicing it as well. And all these combined together, the field is moving on to what is called a big data-based healthcare, where all sorts of data, including lifelog data like pulses and several physical attributes, and genomics data obtained from NGS sequencing, and biomarker data obtained from chemistry experiments, can all be combined to provide you better health benefits as a whole. And our body is constantly affected by several factors such as DNA, environment, and exercise. So by utilizing these factors in healthcare, the world is moving on to more specialized healthcare industry as well. So that ends my presentation on how genomics could affect your lives and how it is being used in the field of research. And thank you very much for your attention. And now I'll be taking your questions if you have any. Thank you very much.
Sir, thanks a lot for your valuable and technical session, sir. It was really, I think it was very much helpful to all the participants out there. Kindly accept our e-certificate as a token of honor from our institute, sir.
My pleasure. Thank you.
Just a moment.
Mm-hmm. Thank you so much. Now, I'll be taking your questions now.
Sir, just a minute, sir. We have to share the certificates to you, sir.
Okay, thank you. Oh my goodness, I'm not a doctor. I don't have a PhD, but thank you so much.
Thank you, sir. Thank you, sir.
Okay, thank you very much.
Sir, participants have raised a few queries through the chat box. May I now request Dr. Sendil, sir, Associate Professor, Faculty of Pharmacy, to join us in delivering the participants' queries.
Yes,
Sir,
thank you.
Sendil, sir.
Yes, madam. Thank you, sir, for your nice presentation.
Okay.
The participants raised some queries.
Okay.
The first question is, how can analyze probiotics by NGS Metagenomics from Vijay Sarkar.
Thank you very much. That's a very good question. Now, this all has to begin with the most basic concept of sequencing. Now, as you know, sequencing is the technology that allows you to study the sequence of an organism, correct? Now, sequencing a metagenome is the same as sequencing anything, except the biggest difference is that in a metagenome, it will contain countless different species of bacteria, strains of bacteria in them. And in order to sequence that, you need to sequence a large amount of data, basically. And by using first generation technology, there is no way you can sequence them all together. But like I said, NGS can sequence a large number of data at the same time. So by getting so much data, you can actually sequence the individual genomes of all those bacterial strains in the sample. So if we move on, like for instance, if this soil sample here contains a large number of bacteria, you can actually extract the DNA of this bacteria combined and just sequence those DNA by getting a lot of sequence data. And after the sequence data is obtained, you can reconstruct them through bioinformatics into individual bacterial genomes. And by that way, you can identify, using the bacterial genome, which bacteria is living inside the actual sample. Does that answer the question?
Okay. Next question.
Okay.
How the whole blood and buffy coat are differentiated in DNA extractions?
Pardon me?
Whole blood and buffy coat are differentiated in DNA extractions or in extracted DNA.
Yes. There are several established protocols in DNA or RNA extraction in the field. And yes, there are specialized extractions for buffy coat blood and also just whole blood. So yes, there are several extraction methods available. Was that the question? Or were you asking about the actual technology? Those two are different, yes.
Sir,
maybe I misunderstood the question.
That is differentiate the whole blood and buffy coat.
Yes, you can differentiate that. But extraction methods are only slightly different. But yes, in principle, they are the same. So yes, there are differentiated methods for buffy coat and whole blood DNA extraction. And I don't have the exact protocol with me right now, but there are several established protocols for them. Yes.
Okay. Thank you, sir. Thank you, sir. The next question is,
okay,
are there any drugs being developed for cancer based on exomes?
Based on exome.
And any potential targets can use it from the Absam Fatima.
Okay.
That is based on exomes.
Yes. short question is yes. I'm looking for, I didn't bring the example with me today, but yes, several hospitals actually have their own medical labs to identify these specialized genes that must be targeted for cancer. And to identify those genes, many employ NGS sequencing technologies. So by sequencing those particular genes of interest, the medical researchers can get information on what sort of mutations are there. And depending on that, they can also devise various treatments. But to be honest, right now, it is more being used in the field of diagnostics rather than treatment, although several treatments are being used and tested even as we speak. The most prominent field of exome sequencing is in diagnostics of the actual cancer. Yes.
Okay, sir. The next question is,
Okay.
how the NGS has revolutionized the SARS-CoV-2 genome sequence analysis in worldwide by Preeta Mukherjee.
Pardon me, I didn't get the question.
They-
How did it revolutionize
Yes,
what?
yes. The revolutions, the NGS has revolutionized the SARS-CoV-2 genome sequence analysis.
Oh, genome sequencing analysis, COVID. Okay, I maybe should have discussed a little bit more about the actual concept of reads. You know, the basics of NGS and all sequencing technology is all about data output. This data output. So, consider that you are reading this book here. First generation sequencing will read one letter, one words at a time, maybe in this kind of speed. But what NGS does is it reads huge amount of data each time it is read. So you can consider that NGS can read everything much faster in a sense. And if the sequencing technology was not revolutionized by NGS, just like in this example, it would have taken so much time to study a genome of any organism, even small organisms like viruses. It takes much longer than, well, and more costly, more expensive than NGS. So it all comes down to the question of how fast and how cheaply can you study an organism. So it's more of a question of economics rather than the actual technology in this case. So basically, NGS can read everything much, much, much faster than any previous technologies in the field. Yep, I think that answers the question.
Okay, thank you, sir. And then the next question.
Okay.
In performing or in transcriptome experiments for bacteria, how to calculate the sample size? In
Genomic, the sample size.
yes,
Okay. Sample size, is that does that mean the sample genome size or number of samples here? I don't think I'm reading myself. Are you talking about sample genome size or number of samples? Okay. How do you consider that? Okay. Well, bacteria is simply, if this is a bacteria, if it's a pure strain bacteria grown in cell cultures, basically, if you harvest the bacteria in the same culture and submit extract DNA together, that will be considered a single sample rather than multiple samples. So each batch would be one sample in that case. But if you're talking about a metagenome sample where you gather it from the environment, so from a soil sample, you'll never be able to tell how many samples are there. So you just sequence them all as one sample and produce enough data so you can actually reassemble the individual sequence using a computer to reconstruct individual bacterial genomes in the sample. So long question short, yeah, one culture batch, one sample.
Okay.
I think that's it. And the question is, yeah.
Thank you, sir.
Okay, I have more. Yeah, I also saw an interesting question there about how do you sequence the epigenetic modifications on DNA like methylation and acetylations? Yes. That's actually requires several techniques that's actually not actually the field of just NGS, but several, the key, excuse me, okay, the key is to, sorry, yeah, okay. The key is to study the individual methylation or modification. To study individual modifications by using specialized reagents or employing techniques like immunoprecipitation to just get the sequences of interest. And Illumina, for instance, has several technologies that specifically targets methylated or acetylated regions in particular. They will specifically target those regions through probes, and after the probes have been attached to those regions of interest, all other DNA sequences are removed, and those remaining sequences captured by the probes will be sequenced. And by getting those DNA sequences that remain, you can tell that those sequences belong to regions that have been modified. That answers the question? Oh, wait, is everyone? Okay. Wait a minute. Is my presentation on right now? Oh, wait a minute. That's okay. That's different.
Hello.
Okay. Sorry, I'm just going to override.
Thank you, sir. Thank you for your nice clarifications for all of the questions.
Okay.
Next,
Okay.
vote of thanks.
Thank you, sir. Thank you, Sendil sir. Thank you, sir, for your valuable question and answer round. May I now request Dr. Hemalata ma'am, Faculty of Pharmacy, to propose the vote of thanks for today's webinar. Ma'am,
Okay.
over to you, ma'am.
Of Faculty of Pharmacy, it's my privilege to deliver vote of thanks. First and foremost, we extend our heartfelt gratitude for our management, founder, chancellor, Dr. A.C.S. Shanmugam sir, Honorable President, Thiru S. Arun Kumar sir, Honorable Secretary, Thiru A. Ravi Kumar sir, under whose benevolence all our efforts are taking shape. Our Vice Chancellor, Dr. S. Geeta Lakshmi ma'am, for the constant support and guidance. I am extremely thankful to our speaker to grace this occasion with a very valuable and informative talk, sir. Our beloved principal, sir, for his guidance and encouragement in all our efforts. Finally, I thank all the participants, students, and faculty members present for this webinar. Thank you all. I request all the participants to fill the feedback link which has been posted in the chat box before today 6 p.m. Thank you for all joining for the session. We'll see you in the upcoming webinar. Thank you all.
Thank you so much for coming to the webinar, everyone. Thank you so much.
Hello, sir.
Yes, hello?
Hello, sir. I want to ask a question.
Yes, please.
Sir, how do you manage to sequence correctly in the midst of all the noise produced in NGS sequencing, sir?
That's another very good question, sir.
Thank you, sir. Thank you so much.
Yes. How should I put this? Yeah, I didn't go into the details of the actual techniques of sequencing today because I heard most people here were not fully proficient in the field of biology. So, okay. I really wish I had a PowerPoint myself.
Sir, can you give me the reference where I can study the answer, sir? Because I'm not able to get any answer for this.
Okay. I should have brought up some backup files. Okay, just a moment, please. Let me just explain this right here.
Thank you. Thank you so much. Thank you so much.
Yeah, I'll just explain this right here with.
Sir, I had many doubts, but I was not able to post all the questions.
Oh no, yeah. Thank you so much. Yes.
And it was a very nice session, sir. Thank you so much.
No, my pleasure. Okay. Okay, can you see this blank screen here? Okay. Let's say this is a strip of genomic DNA. Can you see this well? So NGS data is produced by multiple reads in a certain region. Okay. So, actually, no, wait a minute. I get a better idea. Sir, can you see this clearly? Okay. When there's an unidentified genomic DNA, and that's fragmented into certain sizes, libraries like these will get then sequenced into individual reads of 300 base pairs. But like I said, like you pointed out, in reality, there are several noises like small unmet reds or things that just don't match those details. Now, the first thing we do is we size select libraries into certain sizes, usually in the range of 350 base pairs. Now, after the fragments have been selected for certain base pairs, those things are the first quality control step one. So you only get a certain number of reads. And then in terms of actual sequencing, the sequencer it says 300 base pairs here, but in reality, usually they run it in 301 base pairs and like that. So all these sequenced reads will get, this is to ensure they get certain cycles and certain length in individual reads, and anything that do not match these sequence read length will be considered noises, and they will be eventually filtered out in the process called demultiplication, or actually, Dr. Kumar.
Yes, sir. Yes, sir.
Yes. If this doesn't answer the question clearly enough, can I have your email address? I'll actually, I'll
Please,
try to explain that, in more detail using more professional.
Thank you so much, sir.
Materials. I try to explain this right here with just chalk, but
Yes, sir, but it was very helpful, sir. I'm sending you my email. Thank you so much.
Yes, thank you.
I have posted my email question in the chat box.
Yes, please.
Yes, sir. Did you got my email?
Oh, my email?
My email, sir. I have sent my email at the chat box.
Oh, yeah, please. Okay.
It's ANKEETKUMAR 9530.
Got it. Thank you.
Yes, sir.
Yeah.
Thank you so much.
Okay, sir. Thank you. Thank you so much for joining us for today's webinar, sir.
Okay. Thank you so much.
Thank you, sir.
How do I?
Sir, can you please post your mail ID, sir?
Pardon me?
Yeah, sir.